this talk, as aforementioned, is considering dormancy phenomena in cancer progression, which is more my field of research. And I will speak about this generally, and then I will touch upon two projects that are not quite myself. One is from Einar Gunnarsson and his co-authors, who is actually a PhD student in my current working group here in Minneapolis. And then there's a project by Nikolai Rode, who has been a master student of Anton Bouvier and myself when I was still in Bonn about a year ago. All right, and before I get started, I want to give you like a brief review about what I actually work on myself and what kind of techniques are involved. So my own research is kind of twofold. On a more theoretic level, I'm working on scaling limits of individual-based models of adaptive dynamics, and that is actually pretty similar to what has already been talked about in the last couple of days, for example, by Jochen Blatt and Anders Tobias. And so we're studying the evolution of an asexually reproducing population. Think of bacteria or single cells. And the entire assumption is that this population is not made up of a single type, but is heterogeneous and will adapt to its environment over time. And these pictures here, I like to draw these pictures to summarize the dynamics of a system where arrows somewhat represent changes to the system, and we can have cells reproducing and dying. And the kind of defining factor of these adaptive dynamic models is that you're working with interaction between individuals. So similar to the models that you've seen, there might be some competitive interaction that acts as an increased death rate, could also decrease the birth rate. And then usually there's the potential for mutation and new types being introduced to the population. And the underlying question is that we're starting out with a model that is defined via individual-based dynamics. So we're looking at rates of single cells dying or proliferating. But what we want to derive in the limit is the somewhat macroscopic pictures, where we want to obtain from this the general motion on the space of potential traits or types over time, according due to mutations and selection going on. And for this, then a fitness landscape, or indirectly you obtain a fitness through these competitive interactions, which then defines your somewhat path on the trait space. And on a more applied level, we have been using these types of models to study cancer dynamics. Cancer by itself is somewhat a perfect model organism for these evolutionary models because, well, the defining factor of cancer cells is that they're producing very fast and they have a lot of mutation going on. So within like a short observable time frame, you can actually see evolutionary dynamics taking place, and you also have the increased selective pressure accelerating these evolutionary processes due to treatment usually. And an important factor that actually is now steering towards also these questions of dormancy is that we quite often see resistance to therapy in cancer due to phenotypic plasticity. So our tumor, as mentioned, is not made out of a homogeneous mass of clonal cells, but there is a variety in there, and different cells react differently to the treatment. Right, and so there are quite a few parallels to the dynamics of viral infections. I'm also working a lot on the immune system interacting with cancer cells, which is of course also involved in viral infections. The key difference being that somewhat instead of the virus relying on healthy host cells and needing them to proliferate in a kind of parasite scenario for cancer cells, we have more of a competitive interaction with the healthy tissue where healthy cells and cancerous cells compete for resources. Right. So far for my background. Oh, and I forgot to mention, please do interrupt me at any time. I see you in a very tiny picture, but I'm not able to like read nonverbal cues of people looking confused. So please feel free to jump in anytime if you have any questions. All right. So a little bit about the biological background of cancer and cancer treatments. So first of all, tumors are not necessarily cancers all the time. Tumors just generally describe a mass of abnormally growing tissues, but those can be either benign, which is more or less harmless. So you have an expanding tissue, some lump or mass, but it's not actually doing that much harm to its environment. It's usually staying in one place, so there will be no invasion of adjacent organs or metastasis. And once these benign tumors are treated, for example, surgically removed, they usually don't grow back. But then there's the malignant form of tumors, which are often called cancer. And those are much more dangerous because they invade organs that are surrounding them, therefore impairing their function, and they can also metastasize and spread all over the body. And so generally there's this distinction, but I'm more or less going to refer to tumors and cancers as the same thing in the upcoming slides. And there are a ton of different types of tumors, basically one for every part of your body. And over the last decades, tumor has become the leading cause of death worldwide, which I looked at these numbers prior to this talk and was actually shocked myself. So about 1/6 of all deaths worldwide are due to cancers. And in the US, I found a number that actually about 40% of the population will eventually develop cancer within their lifetime. So as somewhat a consequence of improving medical treatments and prolonging human lifespan, the chances of developing cancer have become very high. So as of that, it has become very important to find effective therapeutic approaches to get rid of those cancers. And for many years, there have been 3 major approaches: surgically removing the tumor, so just cutting it out, radiation, and chemotherapy. And recently, over the last, 20-ish years, immunotherapies have become an important approach as well, where the idea is basically to enhance the functions of the body's own defense mechanisms to treat tumors. And a downfall of many of these therapies, as the surgical removal, the radiation, and the chemotherapy, is that they are usually quite undirected. So if you cut out a tumor, you're also going to harm the surrounding tissue. And radiation can be focused in on certain areas, but it's always going to hurt the surrounding tissue as well. Same for chemotherapy. Like drugs that are harmful to tumor cells will quite often also be harmful to healthy cells as well. So what has become more and more important is to develop targeted therapies that somehow attack cancer cells specifically without damaging these surrounding tissues. And that sounds easy enough and straightforward enough, but it's actually quite difficult to identify these unique features defining cancer cells and differentiating them from healthy cells. So finding these targets for more specific therapies is quite difficult. And many approaches have been connected to this feature of cancer cells of them being highly productive. So quite a few targeted cancer cells work by somehow interfering with the reproduction pathways of the cancer cells. For example, blocking the receptor for epidermal growth factor, EGF, and many other versions of interfering with reproduction. And the goals of these therapies can both be to slow down tumor growth, but also actually induce apoptosis and kill off existing cancer cells. So how does dormancy come into play here? Well, usually these therapies show initial success, and through surgical removal or chemotherapy, we see an initial remission of the tumor mass. But then quite often, after a longer time, and the longer time can be really long in these cases, so for certain types of breast cancers, that can be up to 20 years, where you initially have a therapy success and you think the tumor is gone, but then after a long time, quite often we will see recurrence of cancer and the cancer will grow back, sometimes in its primary tumor site, but often also in different tissues, which is then called a metastasis. And somehow these tumor cells must have escaped therapy. So by genetic sequencing, you can actually tell that these tumors are quite often derived from the original primary tumor. So it's not just bad luck and a second initiation of cancer, but it's actually stemming from the original primary tumor. And somehow these cells escape therapy and manage to stay undetected over these more or less time spans, long time spans. So, and that is what can be considered as tumor dormancy, staying undetected over a longer time. And there are two kind of ways of looking at this dormancy. If we just define it as tumors staying undetected at a low population size, one way of achieving this is so-called tumor mass dormancy, where actually the cells themselves might still be reproducing, but they have somewhat reached a low level equilibrium between reproduction and cell death that keeps the tumor size in check at a low level. And the alternative, which is closer to what we've been discussing over the last days, is actual tumor cell dormancy, where single cells enter a quiescent state. So there is actually per cell a minimal reproduction and death rates. And so these don't just balance out, but actually the absolute rates themselves are very low. And it is assumed that these processes are reversible usually. And just as an example for this somewhat undiscussed case of tumor mass dormancy, we have actually seen this in a project that I've been working on in Bonn, where we study immunotherapy of melanoma, so skin cancer, where on the left here you see the experimental data where in the cases of successful treatment, the tumor seemingly dies out and is undetectable. But in the simulations and mathematical models, we see that it's actually more likely that the tumor doesn't fully die out, but we have somewhat a low fluctuating level here, which just due to the techniques of measuring these tumor sizes is undetected if it drops below a certain size here. And this state of low-level fluctuations is often described as a tumor immune equilibrium. And what's happening here is that the immune cells and the cancer cells basically enter a predator-prey-like interaction where in the presence of their target, the tumor cells, the immune cells proliferate and then kill off the tumor cells, which then, after their target is gone, also the number of immune cells reduces again, which allows the tumor to proliferate again, and so on. And we see these fluctuations at a low level. But since there is still activity going on, there is active reproduction going on from time to time, there is still the potential for mutation. And what actually can be seen is that sometimes after a long time in this immune equilibrium, there will be new types of cancer cells appearing that can then are possibly immune to the therapy and can then escape and start growing again. Right, and if we now, for the purpose of this workshop and comparison to the virus dynamics, focus in on tumor cell dormancy, this is often triggered by the microenvironment of the tumor. So there are many ways that this can happen. For example, there can simply be cytokines, so chemical molecules in the microenvironment that interact with the tumor cells and somewhat either block activating pathways of cell proliferation or trigger inhibitory pathways or even cell death. Another option is that there could be hypoxic regions. So if you look at a tumor mass, the tumor, many models assume the tumor to be a well-mixed entity, but actually we need to pay attention to the spatial structure of the tumor, where if these tumor masses grow, they actually build their own blood vessel systems in order to be to get nutrients and oxygen. And in the center of these tumor masses, resources are often not quite as available, and we see a lack of oxygen in the tumor core, which then alters the metabolic pathways of the tumor cells and reduces their reproduction. So lack of oxygen can also be a cause for tumor cells to enter a less proliferative or dormant states. And dormancy can, so these first 2 points are not necessarily tied to therapy, but dormancy can also be a way for tumor cells to resist therapy, where by changing their phenotype and altering their pathways or receptors that are expressed on the cell surface, as I mentioned, if we're looking at these targeted therapies, those look for defining features of cancer cells as, for example, these receptors. And if the tumor cell alters its appearance and downregulates certain receptors, this can somewhat remove this target of the cancer therapy and therefore cause resistance. And this is actually quite often seen when looking at metastatic cancer cells. So cancer cells disseminating from the primary tumor, migrating to other tissues of the body, and eventually causing a relapse in tumor, where these new tissues often differ from the site of the primary tumor by their microenvironment. And this can initially induce dormant states in the cancer cells where these new tissues are not what the cells, the environment that these cells are adapted to. But then if there is a change in the microenvironment, and these changes can have various causes, a change in nutrition or whatever, and these changing in microenvironments can then later on trigger a delayed relapse of the tumor when these dormant metastatic cells get reactivated. Yeah, and this potential for dormancy is actually quite common in cancer cells. So it does not seem to be a rare feature of cancer cells. And one question that one can ask himself is whether this potential for dormancy comes at a certain price, which is a phenomenon that is quite often studied in cancer resistance, which is called so-called costly resistance, where keeping up a certain mechanism to avoid therapy, and this might or might not be dormancy, comes at a certain price and is more costly to maintain than the normal cell state, and therefore there is a payoff between maintaining these mechanisms and being more resistant to treatment or other disadvantages, environmental changes. So coming from this biological point, there is a variety of questions that need to be answered when thinking about dormancy and understanding it as a resistance mechanism and cause for delay to tumor recurrence, which is we need to understand what are the precise triggers for cancer cells to enter these dormant states, what chemokines or changes in the environment cause these dormant states, then how can they be detected? So if we look at cancer progression, how can we tell if a tumor is actually fully eradicated or if there is still a reservoir of remaining dormant cells that can reactivate at a later time point? Under which conditions can these dormant cells be reactivated or kept from reactivation? I mean, both of these things could be interesting, where you might want to keep the cells in a dormant state because then they're more or less harmless, or you might want to reactivate them to make them more susceptible to therapy again. And coming from all of this, we then need to identify the best targets to manipulate these dormancy mechanisms. And there's also the question of timing, where during various stages of cancer progression, it can be more or less successful to target these dormancy mechanisms. So that's somewhat the general background. And I want to look at two examples now, but before I do that, are there any questions from your side? People look more or less content in the small picture that I get. So I guess I'll move on. So the first project is the mentioned project of Einar Gunnarsson and Jasmine Fu is actually my postdoc advisor now here in Minneapolis. And they have been studying the role of phenotypic switching in cancer drug resistance. And this can, but doesn't have to be associated to dormancy. So generally, their model setup is that we have a type 0 cell, which you can regard as the wild type cell that is sensitive to a drug treatment. So we have cells reproducing at a certain rate, R0. We have cells dying, and then there is the potential to switch between these two states of cancer cells, type 0 and type 1. And these switches could be associated to birth events, but they don't necessarily have to. Importantly, these models here, we just have a linear death rate, so there's no competitive interaction in this model. And the assumption is somewhat that the type 0 cells are sensitive to the drug, so their net growth rate, birth minus death rate, the lambda 0 is negative, but then type 1 is a resistant state. And the question is whether this potential to switch to the resistant cell, whether that can ensure survival of the tumor overall, so of both type 0 and type 1. And you can study this system theoretically pretty similarly to how we've seen this in the talks of Jochen Blatt, where you can study the eigenvalue of the mean matrix, or I've heard various names for this: mean matrix, generator matrix, Jacobian. And if you study the eigenvalues of these matrix, the largest eigenvalue will tell you whether your branching process, which is what we're dealing with here, a multi-type branching process, whether this process is supercritical, subcritical, or at the critical level. And it turns out that the condition for supercriticality in this case can be written in this form, where basically we're looking at the relation between the switching rate and the net reproduction rate for the different types. And to obtain supercriticality, the potential for switching to the resistant type somewhat has to outweigh the potential to switch back to the susceptible type. And this picture here, you can see on the x-axis the switch to the dormant state called mu, and on the y-axis the switch back to the active state. And this line is exactly the critical case for this equation. So where mu is equal to mu times lambda 1 divided by lambda 0 plus lambda 1. So this is a logarithmic picture. This is generally a linear function here, but becomes this curve here on the logistic scale. And we see different regions where, so basically underneath this curve, the tumor can persist, and the branching process is supercritical. And there are lines for two different scenarios where either the growth rate of the resistant type can be of the same order as the decay rate of the susceptible type, or it can be much smaller. And that this second case is probably more close to the picture of dormancy, where the reproduction rate of the resistant cell is actually very small, in particular small compared to the decay rate of the susceptible type. And so in this A region, the tumor can survive simply because the rate of reactivation is smaller than the net reproduction rate of the tumor. So what that basically means is that the cell can divide and produce offspring before it switches back to the active state. So just the resistant population by itself can grow. But then there's also a small region here where this is not the case, but nevertheless the tumor can persist, and that is because so in this case, this mu here needs to be relatively large to balance out the large rate mu, and that means that the loss of resistance is compensated by very fast adoption of resistance. And then we have this large D region where extinction is ensured in the process. And one can further study this. So this is the same X and Y axis. This line here is the same line as before of the critical reactivation rate. But one can now study the probability of the tumor surviving. So as mentioned in the upper region here, we have a subcritical branching process, which is destined to die out. But even if we have a supercritical branching process in this lower region, there's still the potential for extinction simply by chance. And so in this case, they looked at starting out with a fully sensitive population of cells. And then you can really see that depending on the rate of acquiring dormancy, even in this supercritical scenario, the probability of survival, which is what is plotted in this somewhat heat plot here, can be very low or even close to zero. So and that is because of the initial conditions, if we start out with these sensitive cells, basically we need to ensure that dormancy or resistance is acquired before the wild-type tumor can die out. And we see a rather gradual transition here of increasing this probability. So looking at this picture, what they further done in their paper is to study the effects of combining the original treatment with a potential epigenetic drug that modifies these rates of acquiring dormancy or reactivating through epigenetic changes. So the goal is somewhat to decrease the rate mu of acquiring resistance or increase the rate of resensitization or reactivation, if you think about the dormant picture. And they slightly changed their model for this, because in the paper, and I'm not going to talk a lot about this, they're actually looking at a third phenotype here that models the acquisition of stable non-reversible resistance. And so instead of looking at the tumor outgrowing the therapy, what they've been studying is, under different therapy protocols, what is the potential for acquiring this stable resistance, which then basically means guaranteed therapy failure, because you then have a type of tumor cell that can grow and is permanently resistant to the therapy. But the potential for acquiring the stable resistance is nevertheless tied to tumor survival in the original model as well, because we only see this switch from the reversible resistant type. And in this picture, we're now looking at a treatment, the original cancer antigen, having somewhat on and off phases here. And that is because quite often cancer treatments are associated with severe side effects. So you will basically switch between on and off phases of treatment. And the question is now, how should these on and off phases of anti-cancer agents be combined with this epigenetic drug influencing the switch between sensitive and resistant states? And they've tried out a variety of hypothetical protocols where there could be alternating treatment between the epigenetic drug and the anti-cancer agent, or it could be applied partially simultaneously. And it turns out that, so this blue line, you see this zigzag because basically during the, so this plot shows the probability of acquiring stable resistance, and these plateau phases correspond to the off phases of the drug, where most of the cancer will revert back to its sensitive state, and therefore we cannot acquire the permanently resistant type during these phases, and therefore not increasing the probability to obtain stable resistance up to this time point. And we see the worst thing we can actually do is pre-treatment with this epigenetic drug, because the consequence of that will be that before treatment, all of the cells will be in the sensitive state and proliferate very fastly. So I forgot to mention that, but without the presence of the drug, this sensitive cell actually has a much higher proliferation rate than the resistant type, which is in line with our dormancy interpretation. And that just means that all the cells pre-treatment will be in this sensitive state, can expand very fastly, and therefore we somewhat have a good initial condition for treatment onset. And many then under the treatment, we have many sensitive cells that can revert to the resistant state and then trigger this stable resistance. And then simultaneous treatment, or alternatively alternating treatment but applying the epigenetic drug after the anti-cancer agent, that seems to be all pretty similarly and equally promising, where the idea is basically to revert the resistant state back to the susceptible state and therefore be able to treat the cancer again. So as a conclusion of this project, we can see that even reversible phenotypic alterations such as dormancy can save the tumor from eradication. So there doesn't necessarily have to be a permanent resistance mechanism as a full-on genetic mutation of the cancer cells to avoid therapy. We see that the success of therapy evasion is highly dependent on the relation between the rates of acquiring resistance and losing it again, and that epigenetic drugs that alter these rates could be a promising approach in combined treatment, but it's also very important to pay attention to the timing of these treatment schedules and when to manipulate these switch rates. So that is as much as I wanted to say about this first model. I don't know if there are any questions. I went over this very briefly. So if you're interested in details about this model, the paper is actually contains much more content talking about this stable resistance. So I'll make sure to put references to these papers in the slides that I upload. All right. There's no intermediate interruption. I'll move on to the second example. So in this model, two aspects of this model are that we have no interaction or competition between individuals. So it's a pure, I mean, we have somewhat interaction in terms of switching between types, but we don't have competition. So it's a pure branching process, and we have no limitation of resources or anything that would limit our population size. And then also the switches to and from the resistant or dormant state in this model are spontaneous. So they're not triggered by any environmental factors. And that is, both of these aspects are changed in the second example. So I want to briefly remind you of the model of Jochen that has been discussed on Monday, where there were two different cell types, type 1 and type 2, and I have hopefully chosen the same parameters as in Jochen's talk to not confuse you any further. So now lambda is not the net growth rate, but lambda is actually the rate of cell division, lambda 1 and lambda 2 respectively for type 1 and type 2. And we have, in addition to a natural death rate, we also have a quadratic competitive factor, where these rates here are somewhat per cell rates. And then we have this quadratic term of competition, which limits our population size to a certain equilibrium level. And the type 2 individuals under this competitive interaction have the potential that with the probability P, instead of dying, they revert to a dormant state. And this mu should not be here, I just realized. You just have the second term here. They can stay in this dormant state for a while, still have the potential of dying, but probably with a reduced death rate where kappa is smaller than 1. and can reactivate at a certain rate sigma. And we've seen previously that this type 2 can invade type 1 if somewhat the advantage of dormancy, so the probability of acquiring the dormant state and then reactivating before dying, if that outweighs the advantage of the one type due to a higher reproduction rate. Right, and so in a project of Nikolai Rode, a master student of ours, wanted to study, we have this density-dependent switch to dormancy. And so we asked ourselves, well, if this acquisition of dormancy is a consequence of limited resources and competitive interaction, what if the reactivation also is density dependent? So thinking of cancer treatment or also metastasis, what if the reactivation actually depends on environmental conditions, such as having enough space and resources? And so we changed this constant rate sigma into a rate sigma n that depends on the population size, where the combined size of active individuals, so either type 1 or 2a, actually has a negative impact on this activation. And again, we see a rescaling by k here, the carrying capacity, where we assume that this impact here is less and less severe the more resources there are. So if we make this assumption, we look at this new rate in particular, we see that we're only looking at the positive part here. And if the current population size is larger than k divided by this new parameter gamma, then actually the activation rate will just be equal to 0. So if we, in this setup of looking at a dominant type 1 population and the potential for invasion of type 2, if we start out with an equilibrium of type 1, and this is now the rescaled size, if this is larger than 1 over gamma, and we are dealing with a lower reproduction rate lambda for the type 2 individuals, then the switch to dormancy can essentially be viewed as a death because we have no reactivation. And hence, if we start out with a small type 2 population, since it has a smaller reproduction rate, the process is subcritical and the type 2 mutants will never be able to grow. So instead of looking at this scenario, what we wanted to study is the potential of these type 2 individuals that have the option of dormancy to recover, to first of all survive these unfavorable conditions and then recover if eventually there is a change in the environment. And that can be due to treatment intermission or through entering a new niche, a new microenvironment due to metastasis. And so what we're looking at is initially the type 1 individuals are the fit ones, as mentioned above, that will take over the population. We see the type 2 individuals slowly decaying. But then if there is a change in fitness here that alters in particular the fitness of type 1, but probably also of type 2, and the equilibrium state of type 1 drops below this 1 over gamma threshold, then we can have reactivation of the dormant type 2 cells, and they can start proliferating again and might eventually take over the tumor again. So the question is, what are somewhat the criteria for type 2 to survive long enough for this and how fast can the type 2 then recover after we had a change in fitness. And a way of looking at this, I like to, instead of looking at the absolute population size, what we like to study is somewhat the K exponent, so the order of magnitude of the population size, which will eventually be rescaled by K. So beta will lie somewhere between 0 and 1, where if beta is 0, the population is extinct due to this -1. And if beta is equal to 1, we have a population size of order K, which in particular then will trigger these short-term Lotka-Volterra interactions according to the deterministic limit. And the nice thing is, if you look at a log K timescale here, exponential growth of these branching processes that we study for a small population then translates to linear growth of this exponent. And that allows us to draw nice pictures to study the growth and decay of populations, where here on the top is the original picture in terms of population sizes. And on the bottom, we see this exponent beta, where initially, if we start out with all populations, so as a reminder, this somewhat pink is the type 1, the other two are the type 2s, where orange is the active, blue is the inactive state. So in the initial phase, if all populations have a substantial size of order K, as mentioned, if we start out above this critical threshold for suppressing reactivation, one can actually show that we will stay above this threshold, and hence we're essentially looking at a 2-type Lotka-Volterra system, and type 2 is the subcritical type in this case, and the Lotka-Volterra system will just yield the equilibrium of type 1 being at its monomorphic equilibrium and type 2 dying out. And so type 2 will drop below a small epsilon threshold pretty quickly in a time of order 1. And after this, we can now study the microscopic decay of this type 2 population, which takes place on a logarithmic timescale. And since the growth rate of the active type 2 cells is negative, this yields extinction in logarithmic time, and actually these the slopes in these pictures here correspond exactly to the exponential growth or decay rate. So here we will have a slope of λ2 minus λ1 in the picture. So drawing these pictures, that's really actually quite helpful in determining of what will happen in such a population. So we see that the active individuals will eventually die out. We can regard them as a monomorphic branching process because we have no reactivation from the dormant cells. And the dormant cells have two options depending on the parameter regime. If they have a relatively high death rate, then they will just tag along with the active cells because they get on the timescale of order 1 input from these active cells due to this switch to dormancy, and so they will tag along at the same population size. However, if the death rate of the dormant cells is relatively small, they will be able to survive longer than the active cells because they don't get this input anymore from active cells becoming dormant, but their death rate is actually smaller than the net growth rate of the active individual. So what can happen is that this dormancy then allows for a longer survival of the type 2 population. And if at this point we now have a change in fitness, the dormant state might have survived long enough. And now if in a time of order 1, again, the type 1 individuals reach a lower equilibrium that is below this critical threshold, that will yield now a positive reaction rate, a reactivation rate sigma, and the type 2 individuals can reactivate. And we will see an immediate jump of the active type from 0 to the same order of magnitude as the dormant cells. And if we assume that the net growth rate of the type 2 cells is now larger than the one of the type 1 cells, well, of course, now then the type 2 population will be able to invade the population and take over. So the question is, even if we make this assumption that type 2 is now, even without dormancy, fitter than type 1, is there still any additional advantage of dormancy apart from having the population survive long enough to witness this change in the environment? And the answer is yes, because dormancy still will allow the type 2 cells to recover much faster. For once, because due to reactivation on the time scale of order 1, we have a vast influx of active cells here initially. So within a very short time, the active cells will have the same order of the population cells as the dormant cells, which somewhat gives them a better initial condition for growth. And then, as we've seen on Monday, if you look at the actual growth rate of the type 2 cells, which can be found by looking at these eigenvalues again, this also turns out to be strictly larger than just the growth rate of the active type 2 individuals by themselves. So we both, if we look at the time to invasion of the type 2 cells, we both get a higher initial condition here, which somewhat decreases the distance that we need to make up for here. And it also increases the rate of growth, so the slope of these lines, to arrive at a population of order K again. Right, so the conclusion here is that even if cells with the potential for dormancy are unfit under normal circumstances, because, for example, this resistance, upholding this resistance mechanism might be costly, environment-mediated dormancy can still be beneficial to cancer cells, both by slowing down and preventing extinction under these unfavorable circumstances, and also by allowing for faster recovery, both due to a larger initial reservoir of cells to grow from, and also by an increased growth rate. And I see we're nearing 4 o'clock. That is perfect, because I'm now coming to my last slide. So based on these examples and many other things, I have a long list of open questions considering dormancy in cancer, but I think there are many questions that are actually aligned with studying viruses. So first of all, an important question is how, if we're looking at experimental data, is there a way to distinguish between these two types of dormancy, the tumor mass dormancy that I mentioned in the beginning, where we just have a low equilibrium size, but still proliferation and cell death going on, versus tumor cell dormancy. And this is a question of model selection, but it's actually also important when you think about mutations, because if a cell is in a dormant state, there is not much reproduction and not much mutation going on. But if we're actually dealing with a low equilibrium at a tumor mass dormancy, this still bears the potential for mutations and acquiring new genotypes that could be permanently resistant. it's important to study what mechanisms and environmental cues actually trigger dormancy and reactivation. As a consequence, we need to think about which parts of this microenvironment actually need to be modeled when deriving these biological models. So which parts do we actually need to depict in our models? And in the example that we just looked at, we have a somewhat linear dependence of activation and dormancy switch depending on the size of the population, quadratic for the overall population, but linear per cell. But this could also be a threshold effect, or there could be some saturations going on. So this doesn't necessarily need to be a linear dependence. We can think about the payoff of dormancy, so these costly resistant questions. And then we need to think about what treatments are best. And as I mentioned, in any applications, we will have usually not a continuous treatment, but phases of off and on treatment. And this leads to mathematical questions of fluctuating environments where the sign of the fitness varies over time. And then it becomes a question of comparing timescales in mathematical models where we don't just have the timescale of order 1 of the Lotka-Volterra dynamics, or this log timescale of growth, but we get an additional timescale in which these fluctuations happen. And that is actually a question that I'm currently studying, not in the context of dormancy, but generally in these adaptive dynamics models with Manuel Esser, a PhD student in Bonn. So we're thinking about these fluctuating environments where we don't have a stable state of our system. And lastly, this dormancy can be seen as a mechanism that causes delay, for example, under these metastases. So that is also an interesting feature to study in mathematical models. So this is somewhat a list of questions that I'm thinking about. Hopefully some of those align with the questions that you're asking yourselves. And we'll have the potential of working on those when I join you next week. So that is where I'm done for today. Thank you for sticking with me, and I'm looking forward to seeing you in person next week.